Good morning. So in this class, I am going to continue the same carbohydrate metabolism. So in last class, we have seen some of the basics concept, particularly the introduction about the carbohydrates and its metabolic pathway, different metabolic pathways. So particularly in last class, we have discussed about the important pathway glycolysis. We have seen the glycolysis. So in this class, I am I am going to explain about the next cycle. It's a TCA cycle. It's otherwise is called as citric acid cycle. So the what is TCA cycle? And the TCA is nothing but it's a short form of tricarboxylic acid cycle. Simply we can say it's a citric acid cycle. Otherwise we can say Kerb cycle. So different names is there. So TCA means tricarboxylic acid cycle, otherwise citric acid cycle, otherwise Kerb cycle. Okay. So before we are going to see the exactly what is TCA cycle, we must to know that the TCA cycle it's just a continuation process of uh, carbohydrate metabolic process. We know that in last class we have seen glucose metabolism process. The glucose is first is converted into the pyruvate in this glycolysis pathway. So the glycolysis is a first step pathway. The glucose is converted into the pyruvate. During that process, it may undergo the various reactions. It may produce some energy. It gains some energy. So many processes happen. At the end of the glycolysis, is converted into pyruvate. With the presence of lactate dehydrogenase and oxygen supply, it's may based upon the oxygen conditions. Sorry, last step based upon the oxygen availability and the presence of lactate dehydrogenase enzyme, the pyruvate it's converted into the lactate. We know that these are entire things is comes under the glycolysis. So what's happened after that? What's going to happen once it's pyruvate or lactate is formed means what is going to be the next step? So the next step is TCA cycle. That means citric acid cycle. So I can give one picture. So this picture is exactly give the relations between the glycolysis and TCA cycles. You can see here once you can see here once this is a glycolysis process. Once the pyruvate is formed, then it's enter into the TCA cycle. So in between it's having some other processes there. That means exactly the pyruvate, otherwise lactate interchange. From this pyruvate, it's converted into the acetyl CoA. We know that acetyl CoA is one of the basic raw material, it's energy resource, and it's a best intermediate product for various pathways. So here also the same, the acetyl CoA is a intermediated product to go for the next step. That means citric acid step. So once the glucose is metabolism first process is converted into pyruvate first. This is the first process. The second one is the pyruvate is converted into acetyl CoA. That is second. Then this acetyl CoA is exactly is enter into the TCA cycle. So the TCA cycle is nothing but it's a simply it's oxidation process of glucose. Okay, that's all. So you can see exactly what is happening here. How the intermediate product is formed. That means from the pyruvate, how the acetyl CoA is going to form. This picture is clearly representing that process. You can see here this is the exact mechanism of pyruvate dehydrogenase complex is working. So once this pyruvate, it's converted into the final product of acetyl CoA. This is the first substrate. Then it's converted into the product. This is the exact process. Here, what's the thing is there various enzymes is involved. So first this pyruvate, it's converted into the hydroxyethyl TPP. The TPP is going to carry this ethyl group. So it's converted into the hydroxyethyl TPP. Then next, with the help of oxidized lipomide, this hydroxyethyl TPP is going to, it's a cycle. It's going to donate this hydroxyethyl group towards this lipomide. It's going to form a acetyl liposamide. You can see here, this is oxidized liposamide. So this hydroxyethyl group is going to donate in this group. So it's going to conjoin together. It's going to form a acetyl lipomide. Then with presence of coenzyme, this acetyl CoA plus coenzyme is conjoined together. It's going to form a acetyl CoA. This is the process. Once again, this liposamide, it's a hydrolyzed form, so it's once again is going to oxidize with the help of this process, FDAD and FTH process. It's may converted into further oxidized lipomide category. It's ready for next cycle. So this is a continuous cycles. That means continuously whenever the pyruvate is formed during the glycolysis process, immediately is going to convert it with this acetyl CoA with this help of enzymes. Okay, this is the process. So glucose to pyruvate, first step, then pyruvate into acetyl CoA. That's the second step. Once this acetyl CoA is formed, now the uh, cycle is next cycle is B is going for, it's going to sorry, once this pyruvate is converted into the acetyl CoA, it is ready to enter in next cycle. The cycle is nothing but it's a TCA cycle. Okay, so this is the overview of Krebs cycle. That means TCA cycle. You no need to confuse about this entire concept. No need to write all those things. I will explain each and every step in forthcoming slides. You just see that this is an overview slide. So you simply imagine the two molecules of acetyl CoA. It's enter into the TCA cycle. It's converted into the various products, particularly the four products. One is citrate, then second one is alpha keto glutarate, then succinyl CoA, then oxaloacetate. The acetyl CoA is raw material. The intermediate products are 4 citrate, alpha glutarate, alpha keto glutarate, then succinyl CoA, then oxaloacetate. So in between, these are the 4 major intermediate steps. In between also so many steps is there. You can see here this right side picture. This acetyl CoA, it's enter into pathway, it's converted into the citrate, then isocitrate, oxalosuccinate, alpha ketoglutarate, and succinyl CoA, succinate, fumarate, malate, then finally it's converted into the oxaloacetate. So during that process, it may release more amount of energy. It's release the water molecule and hydrogen carbon dioxide, then it's get the ATP energy source. So during the process, it may release more amount of energy and water molecule and carbon dioxide because it's a glucose oxidation process. So it release more amount of carbon dioxide and water as an intermediate product, and major source is energy compounds. So this is the overview product. So acetyl CoA, it's enter into the cycle. During the cycle, it's going to produce the energy. That's all. Intermediately is more four major compounds is going to synthesize: citrate, alpha ketoglutarate, then succinyl CoA, then oxaloacetate. These are the 4 major things. So in bracket they have mentioned how many carbon atom is there is going to form. So in you can see here, this is a conjoined picture because it's having a structure come naming wise it's mentioned. So you can see here, this is a pyruvate, then acetyl CoA, 2 carbon molecules of acetyl CoA. The pyruvate, it's converted into the acetyl CoA. Then this acetyl CoA is ready to enter this TCA cycle. So the the 2 molecules of acetyl CoA is going to conjoin together. Then the coenzyme is released. Then it's converted into the citrate. Citrate is you can see here, this is a 6 carbon molecule. So it's enter into the various process, cis-aconites and isocitrate, alpha diglutarate, succinyl CoA, succinate, fumarate, malate, and oxaloacetate. You can see here the 6 carbon atom molecule, it's converted into 4 carbon atom molecule in this succinyl CoA. So once the citrate is converted into the D-isocitrate, then alpha ketoglutarate. In alpha ketoglutarate, it's converted into 5 carbon atom. So here 6 up to citrate to isocitrate, it's a 6 carbon atom. And alpha ketoglutarate during conversion is converted into the 5 carbon atom. In succinyl CoA, it's converted into the 4 carbon atom. So this 4 carbon atom molecules is further converted into the succinate, fumarate, malate, and oxaloacetate. So during that process is going to produce more amount of energy. You simply memorize that 6 carbon atom, it's converted into the 4. The 4 carbon atom molecule is going to convert into the various process. During the, it's going to produce more amount of energy and oxidation is going to happen. So this is structure wise. You can see here structure wise explanation. So the Yeah. Two acetyl-CoA molecules, two acetyl-CoA enzyme molecules is going to conjoin together. It's going to form a citrate. Then citrate, it's converted into the isocitrate. It's an intermediate product. Intermediate form is cis aconate. Guys, I wanna tell you another one thing is you can see this overview slide. In this TCA cycle, most of the reactions it's a reversible reactions. In some of the reactions only it's a irreversible reaction. You can see here acetyl-CoA to convert into citrate, it's a irreversible. From the citrate to isocitrate, it's an interconvertible reaction. Citrate is converted into the cis aconate, then it's further converted into isocitrate. So the citrate to isocitrate form, it's an interconvertible reactions. So it's may reversible. That means it's a reversible reactions. Once it's converted into isocitrate, the same isocitrate maybe it come back into citrate form. The same manner, most of the reaction here also isocitrate to oxalosuccinate, it's a interconvertible reaction, and up to alpha ketoglutarate, it's an interconvertible reaction. But alpha ketoglutarate to succinyl-CoA, it's a single reaction, it's not a reversible reaction. After that, once the succinyl-CoA is going to form, that means 5 to 4. Okay, this is 6 to 5 carbon molecule. Once 5 to 4 carbon molecules converted up, once the succinyl-CoA is going to form, it's converted into the oxaloacetate. This entire reactions are interconvertible reactions. That means it may enter into the forward reaction also. Otherwise, it may, if it is during any conditions, it may go for reversible reactions also. This is important thing, because in this reactions they never indicate it's a reversible or irreversible. We just memorize that. So the citrate, it's first converted into the isocitrate. It's intermediate product is cis aconate. So structure wise, what's exactly happening and all I have explained in next slide. So isocitrate, this isocitrate, it's converted into the alpha ketoglutarate. So you can see here this acetyl-CoA enzyme, it's converted into the citrate with the help of citrate synthase enzyme. That enzyme is responsible for the formation of citrate. In second stage, you can see here this aconatase enzyme. Aconatase enzyme, it's responsible for conversion of citrate into isocitrate, because it's going to act on both the stage. That means it may act on the intermediate product also. So this enzyme is converted into isocitrate. The next one is the isocitrate is down regulated into the ketoglutarate, alpha ketoglutarate. So the dehydrogenase enzyme, isocitrate dehydrogenase enzyme, they're responsible for this conversion. That means the isocitrate is converted into alpha ketoglutarate with the help of isocitrate dehydrogenase enzyme. The next, this alpha ketoglutarate once again is going to dehydrate, then converted into succinyl-CoA. So the enzyme responsible here is alpha ketoglutarate dehydrogenase enzyme. So in this both the reactions, you can see here the carbon dioxide is going to relieve. That means in structure wise, you can see here the CO2, that particularly this molecule is going to relieve. So it's converted into the succinyl-CoA. You can see here this reaction, the isocitrate to alpha ketoglutarate also. The second carbon molecule CO2 is going to eliminate, so it's converted into the alpha ketoglutarate. In second step, this carbon dioxide is going to release, so it's finally is converted into the succinyl-CoA. Then with the help of succinyl-CoA synthase enzyme, the succinyl-CoA is converted into the succinate. Here one phosphorylation group is going to, it's act as the energy resource. So the GDP is get converted into the GDP, because during the reaction the energy is going to release, so that energy is gained by GDP, guanine diphosphate, then it's converted into the triphosphate. Then the succinate, it's converted into the fumarate with the help of succinate dehydrogenase enzyme. Once the fumarate is there, means with addition of water molecule with the help of fumarase enzyme, the fumarate is converted into malate, L-malate. Then finally the L-malate is converted into the oxaloacetate with the help of malate dehydrogenase enzyme. So maybe in this structure wise, what's the difference exactly happen? Those things I have explained with this animated slide. So you can see here, this is an entire cycle. It's denoted in a different color representation. So this is the entire reaction. So you can see here from first things. So this is the exact your first starting material. That means pyruvate. Otherwise, it's the end product of glycolysis. The glucose is it's converted into the pyruvate. So once the pyruvate is there, it's converted into the acetyl CoA with the help of pyruvate dehydrogenase enzyme. Actually, this is intermediate step. So the glycolysis TCA cycle is the next step. The intermediate step is this one. The pyruvate is converted into the acetyl CoA with the help of pyruvate dehydrogenase. So what's the structural difference we can see here? Nothing difference. Only this oxygen molecule has been replaced by this coenzyme. So CH3CO and CO, this oxygen molecule it's removed. Then coenzyme has been attached at this place. So it's the carbon molecule and the oxygen molecule is relieved as a carbon dioxide. So this is a exact process happening intermediate step. So after that, this acetyl CoA is now ready. Now is ready to enter into TCA cycle. So with the help of citrate synthase enzyme with presence of water molecule, it's converted into the citrate. You can see here two molecules of acetyl CoA. Two molecules of acetyl CoA is going to involve. So you can see here two molecules of acetyl CoA with presence of hydrogen water molecule. So it's converted into the citrate. No difference at all. The same thing you can add another one CH2CO because it's a two molecules with the presence of one more two hydrogen ions and oxygen molecule. It's converted into the citrate. So the citrate is converted into the isocitrate form with the help of actinidase enzyme. So it's an interchangeable reactions. So what is the chemistry wise difference means nothing. The hydroxyl group, you can see here this hydroxyl group, the second position hydroxyl group is exchanged into first one. That's a difference. So this is first, this is second, this is third carbon atom. So the second hydroxyl group it's shifted to the third carbon molecule. So it's converted into the isocitrate. So the same thing, once you have write the basic core structure, only the structural rearrangement is going to happen. So you no need to mug up entire structure. Once you know the core concept structure, otherwise basic core molecule means from that the structure is going to simply modify. Okay. So the citrate from the acetyl CoA to CH2CO, then intermediate one molecule is there. So totally 3. So the second hydroxyl group is shifted to third position. So it's called as isocitrate. Then this isocitrate it's further is converted down related to alpha ketoglutarate. So the enzyme responsible one is isocitrate dehydrogenase. Okay. So here you can, what is the thing is there exactly happen? The same thing, CH2CO is same. Here the carbon dioxide is removed. That means this CO2 is removed. The second position CO W bond is there. So that CO2 is removed. So it's CH2 CH is there. So with the presence of donation of hydrogen molecules. So you can see here this hydrogen is converted into two hydrogen is there. So this two hydrogen is going to join together. So CH2 COO CH2 is formed. So once again it's going to convert as here CO and COA. So it's isocitrate, it's converted into the alpha ketoglutarate. The next one is with down related. This alpha ketoglutarate, it's once again is converted into the succinyl CoA with the help of alpha ketoglutarate dehydrogenase enzyme. Here, what is the structural difference is there means nothing. This CO group, once again, the same thing is the CO2 is eliminated. So coenzyme is going to add. So instead of CWO, the acetyl CoA is going to coenzyme is going to join together. So it's going to form a succinyl CoA. The next is converted into succinate. The succinyl CoA is converted into succinate. So here the acetyl enzyme is coenzyme is going to remove here. coenzyme is going to remove. So energy is going to produce the one phosphorylation is going to happen. So the phosphorylation taken into GDP. So the GDP is converted into GTP, guanosine triphosphate. So GDPs get phosphorylated is converted into the triphosphate. It's converted into triphosphate. So the structure wise washing, what the difference? Nothing is difference. Only you have to remove the coenzyme. So the structure is become CH2CO and CH2 and COO. So now the succinate is converted into fumarate. Once again, the dehydrogenase is going to happen. So with the help of succinate dehydrogenase enzyme, it's converted into fumarate. So here, what's the structural difference is means the same. One hydrogen has been removed, so it's CHCOO. Then here also it's converted into CH and COO. So two hydrogen atom, here one hydrogen atom, here one hydrogen atom. The two hydrogen atom has been released. So that FDH is taken up the two hydrogen atom, it's converted into FADH2. So the two hydrogen atom has been removed from succinate, then it's converted into fumarate. So the next step is fumarate, it's converted into malate. So what's happened here? Here with the addition of water molecule, so here the hydrogen is going to happen. The structure wise, what's the difference means? One hydrogen, the H2O, one hydrogen atom is going to attached in this position. One hydrogen is going to attach in this position, so it's converted into CH2COO. Then another one, OH group, that means in H2O one hydrogen is attached second position, then OH is going to attach on first position, so it's converted into malate. So the next, finally the malate also is converted into oxaloacetate with the help of malate dehydrogenase enzyme. So what's the structural difference here? You can see here also the same thing. The hydrogen atoms is going to remove. Structure wise, you can see here. So the H2O is going to remove. So CO here you can replace the entire OH group and hydrogen is conjoined together. It's released here. It's hydrogen is going to remove, so it's converted into CWO. Oxygen is remain on the position. Only the two hydrogen atoms is going to remove. So the two hydrogen atom is taken up by the NAD. It's converted into NADH plus and free hydrogen ions. So finally, it's converted into oxaloacetate. So this is your entire reaction, TCA cycle reactions. So this is the next slide. Why this TCA cycle is very important? Because it's one of the important oxidation process of glucose. So in this citric acid cycle, we said various intermediate product is formed. Particularly, the glucose is converted into acetyl CoA. That's the first process. Then it's converted into the citrate, alpha ketoglutarate, then succinyl CoA, malate, oxaloacetate. So these are the 5 products. It's may responsible, otherwise may involve for various reactions. That means from that product, it's may converted into the some other, enter into the some other reactions. So this slide is going to explain the exact pathway. So major synthetic and analoprotic pathway for intermediates of citric acid cycle. Yeah. So first we can go for acetyl. Acetyl CoA is converted into the citrate. That's the first process. So the citrate is may involve for the synthesis of fatty acid and sterol. So this is an intermediate product, we know that. So the citrate, it may from this place itself, it may enter into the fatty acid and steroid synthesis also. Otherwise, once the process is going to continue, the citrate is converted into the alpha ketoglutarate. Means from this place also, the alpha ketoglutarate, it's may responsible, otherwise may convert into the glutamate. That means transamination is going to happen. Once its transamination is going to happen, means it's converted into the glutamate. So the glutamate, we know that it's one of the important things in the brain level. So apart from that, the glutamate, it's one of the non-essential amino acids and purines. So it's going enter into the various pathway. So from the alpha ketoglutarate, it's undergone the transformation process for the glutamate production. Otherwise, the alpha ketoglutarate once again is continue the pathway. It's enter and converted into the succinyl CoA. Means this succinyl CoA is responsible for the heme production. We know that what is heme and all. Hemoglobin basic raw material is heme. So for that synthesis process, the succinyl CoA is going to involve. Otherwise, the cycle is going to continue. The succinyl CoA may converted into the next product, malate, fumarate and malate. So malate means with the help of malic enzyme, it's once again is converted into the pyruvate. So the pyruvate means is a back reactions from the malate. It may enter into the pyruvate. So it's responsible for the gluconeogenesis process. So during the gluconeogenesis process, during gluconeogenesis process, the glucose is going to synthesis from non-carbohydrate material. So the from the malate, it's converted into the pyruvate. From the pyruvate, it may enter into the back process. So that's a thing. Otherwise, the malate is once again is converted into the oxaloacetate. Means the same thing here. Here also, it may enter into the two reactions. With the help of pyruvate carboxylase enzyme, it's once again is converted into the pyruvate. Otherwise, in the transformation process happen means it's converted into the aspartate. Aspartate is one of the basic raw material for the purine amino acid sequence and amino acid preparations. So these are the entire explanation for the intermediate product responsible for various pathways. So that's all. So here itself I can wind up this session. So that's all.